This episode of Cell and Gene, the podcast, is brought to you in partnership with Thermo Fisher Scientific, a pioneer in the development of groundbreaking plasmid solutions. For labs seeking to scale up plasmid purification, Thermo Fisher recommends the Kingfisher Plasmid Pro Maxi Processor, a fully automated benchtop instrument with a press-and-go design that eliminates setup, centrifugation, and pipetting, reducing errors in sample preparation. Additionally, the Mag Max Pro high-throughput no-spin plasmid purification kit automates lower-volume plasmid DNA purification, saving time and enabling scalability. Trust Thermo Fisher to streamline and enhance your plasmid purification processes. Hello, everyone, and welcome to this episode of Cell and Gene, the podcast. I'm your host, Erin Harris, and I've invited Lucas Harrington to be my guest on this episode. Lucas is the co-founder and chief scientific officer of Mammoth Biosciences, the Brisbane, California-based biotech that's focused on leveraging its proprietary ultracompact CRISPR systems to develop potential long-term curative therapies for patients with life-threatening and debilitating diseases. And we'll get to all that in just a minute. But before we do, Lucas, welcome to Cell and Gene, the podcast.
Thanks. Yeah, thanks for having me.
Absolutely. So, all right. This is really incredible information. So you co-founded Mammoth Biosciences with CRISPR pioneer and Nobel laureate Jennifer Doudna, as well as Trevor Martin and Janice Chen. So that's unbelievable, and I can't wait to kind of hear the story behind that. So please do for us briefly explain Mammoth Biosciences as well as the company.
Yeah, so Mammoth was founded back in 2018, and back around that time, just kind of give some of the general history. Right, big breakthrough with CRISPR was made in 2012. This system Cas9 that Jennifer and Emmanuel had originally reconstituted and started to develop was just really still starting to come onto the scene, and a lot of companies were starting to get founded around that. But what we recognized was that there was a large amount of untapped opportunity in terms of the diversity of CRISPR. So CRISPR, I think even to this day, a lot of people think of it as this singular thing, right, where it's basically Cas9 and it's this pair of molecular scissors. But what we recognized was in nature, which is where these systems ultimately come from, there's actually hundreds of thousands of different flavors of CRISPR. And so our idea, and this was originally back when we were doing our work with Jennifer in the Doudna lab, we said, okay, there's probably some unique features in that diversity, and maybe we should take a closer look, not at all 100,000 obviously, but at a few select examples there, and see what are there some of the features that we actually need to develop new therapies beyond what Cas9 can do. So again, this back in 2018 is when we founded the company, but a lot of this work started much earlier, around 2014. And one of the features that we very early on honed in on was just the size of the CRISPR systems, and we can get into more detail on that. But one of the big challenges of the field has been, how do you actually get CRISPR to the tissue of interest to ultimately treat the diseases? So yeah, the company was now founded about 6 years ago. We're around 150 people now, and Jennifer continues to be a big supporter of the company, and it's really a pleasure to be able to work with her for the last decade now, and just how supportive she's been of not just the early basic biology work around CRISPR, but also about how do we actually make this have real world impact? Because I think that's in academic circles, right? There's often people that are just focused on the discovery side of things, and that's great, and that's serves an important purpose. But she really has made a big effort to actually showcase the actual impacts that these different technologies can make.
That's excellent. And it was really interesting to once I was researching the company more, I realized that you and your colleagues had co-founded it with her. Well, quite honestly, I couldn't wait to talk to you. So thanks for all the background information. I'm sure listeners are really interested to get into. Before we kind of get into the nuts and bolts of what Mammoth is doing specifically, I wanted to talk a little bit about more generally and get your perspective on, in vivo delivery, really does still continue to be a bottleneck with, against the clinical advancement of gene editing medicines. And I was hoping you kind of talk us through the why behind that.
Yeah, absolutely. And this is really the problem that motivated us to do a lot of the early work that we did. So happy to go into more detail. But yeah, so gene editing, CRISPR in particular, its ability is really in that it can go and change the DNA in a cell. In a ex vivo context, which is how a lot of these therapies have been done so far, where you're taking the cells out, editing them, and then putting them back in. That's pretty straightforward, right? There's no real delivery problem. You just can very directly insert the material into the cells. In vivo, there's been a lot less actual advancement by the existing CRISPR technologies. A few select examples, in particular targeting the liver, right, where we have non-viral delivery LNPs similar to what you might use for vaccines, where you're actually going and changing the hepatocytes in the liver to make those genetic changes. So that seems to work fairly well. But obviously, the vast majorities of diseases are not caused by mutations in liver cells. And so it really has been the longstanding challenge in the field. And I think if you ask 10 different people from the CRISPR field, I think every single one will say delivery is the biggest challenge that we're facing still. And it really is about how do we safely get things to tissues like the muscle, like the heart, like the brain. And so, I think when we think about the delivery technologies that we have, and this isn't just for CRISPR, but really for cell and gene therapy more broadly, it really comes down to LNPs, which I mentioned, and then viral delivery in particular with AAVs. And I think AAVs they're not perfect by any means. I think anyone that works with them is well aware of the challenges with using AAVs. But that said, they are some of the most robust tools that we have to get gene cargo to cells of interest beyond the liver. And so what we really started, the company on was how can we actually take the fact that we have, technologies that work well now to deliver things beyond the liver, and be able to use those with gene editing. And the biggest limitation from a gene editing perspective is just the size of the cargo. So AAVs, of course, relatively constrained in terms of what you can fit inside of a single AAV. And with the original CRISPR systems like Cas9, they're simply too large to fit into a traditional capsid. And so what we did was to look at this diversity of CRISPR that we mentioned earlier, and go and find and discover and spend a lot of work optimizing and testing those systems to get much smaller CRISPR systems to work very robustly, as robustly as the much larger CRISPR systems that are out there. So the different platforms that we work with now range from about a third of the size of the traditional Cas9 systems to about half of the size. And so really making it practical to deliver not just the CRISPR system with an AAV, but also all the other elements that are required to deliver a gene editing therapy. Because I think that's an important thing to point out as well is, of course, personally, I get very excited about gene editing, but that really is just one piece of the puzzle, right? You need the delivery vector, of course, to help get it to the cells of interest. Also, all the regulatory elements, the gene of interest, I really think of as part of the therapy as well. And so really thinking about, how do we actually think about this from a holistic perspective, as opposed to just thinking about, the gene editing component. And I think that is unique. A lot of people have basically thought about the delivery problem, which is what we often call it in the field, as a delivery problem. And I think we kind of looked at it a little bit differently and said, okay, how do we actually make the cargo work with the delivery that we have already? But we're super excited about what this hopefully can open up for the field more broadly, and get gene editing outside of just the liver and outside of ex vivo therapies, which is really exciting in terms of the progress that's been made. But there's just so much more in terms of genomics and genetics to tackle.
Yeah, absolutely. You made really good points there. And I'm hoping that we can kind of pivot to this next part, in terms of like the editing. I know, having researched you, you guys use new editing modalities beyond the double-stranded breaks, including base editing, which we cover on Cell and Gene quite a bit, epigenetic editing, and more. And so I was hoping you could talk us through why this is and maybe briefly explain how Mammoth is using it.
Yeah. So yeah, I think that's been a really exciting advancement in the field over the last, I'd say 5 or 6 years, really in earnest, is not just using CRISPR as this double-stranded break tool, but actually thinking about it as a way to search out sequences of interest in the genome. So we really focus on 3 different editing modalities, other than double-stranded breaks in particular. So that includes base editing, as you mentioned, RT editing, and then a newer area that I think has garnered a lot of excitement is epigenetic editing. And I think these are all really, really exciting tools. I think honestly that I think there's a lot more to be done as we think about gene editing as a kind of molecular search engine, right, where you can just go and program it and it finds a sequence of interest. I think these 3 that we're focused on right now and have been talking about more publicly are really just the starting point, of this concept. But what we really focused on is how do we actually, going back to kind of the delivery problem, how do you actually get these tools which make that delivery problem honestly much, much worse, right? You're now taking that very large vectoral protein, you're fusing on a bunch of other large proteins, reverse transcriptases or demethylases, and making that much, much more challenging. And so what we've really focused on is how do we take those different editing strategies and actually not just make the CRISPR component smaller and more efficient, but also the fusion proteins that we're putting onto them, and being able to target again tissues outside of the liver, in the muscle and brain are really the focus that we're interested in currently. But yeah, I think it's also been important for us to be able to have. So most companies are focused on just one of those editing strategies, right? And I think that leads to this classic dilemma of kind of hammer and a nail, where you end up driving the pipeline based off of the technology as opposed to based off of the disease biology. And in reality, many times multiple modalities could be used to treat a specific disease. So if you think about, even if you're doing something like a knockout, you could use double-stranded breaks and just make a cut. You could also use base editing to insert a stop codon. You could do the same thing with RT editing, or you can epigenetically silence it. And so for us, it's always been important to be able to look at that portfolio of editing strategies and the disease biology and the maturity of the technologies, and say, okay, which one's actually going to be most suitable for the target of interest. So again, that's been something that's been very important to us, just from kind of a philosophical sense almost as a company, is really being driven by that disease biology as opposed to by the technology.
Gotcha. Okay, thank you. All right, I want to talk a little bit about the studies that Mammoth has going on, and specifically the ones that Mammoth covered at ASGCT's recent. So, as I understand it, you shared data on your proof of concept study on the ultracompact CRISPR systems that we talked about. Talk us through a little bit about the data that you shared at the event.
Yeah. So we have the program that we talked about in particular at ASGCT is targeting a gene called APOC3, which you may be familiar with from other programs, especially with ASOs that try and knock that down and are entering into phase 3 currently. So this gene is tied to triglyceride metabolism and lipid metabolism more generally. And so what we aim to do is to knock that out, to be able to see a reduction in triglycerides, especially for patients that have very, very severe high triglycerides. So this wouldn't be, for for someone that just has moderately high triglycerides. But these patients, when you look at their serum after you draw it, it's almost milky color because the lipids are so high. And so what we're aiming to do is do that in a durable way. So not just having to have an injection every month or every quarter, of actually having this as a one-time curative therapy. And so the data that we presented was in particular focused on both work that we've done in NHPs as well as in our mouse models for hypertriglyceridemia that are humanized for this APOC3 gene. And maybe to just point out a kind of a few of the highlights. So with our CRISPR systems, and this is using a CRISPR system called Cas9, which is about half the size of Cas9. That system, we were able to knock down the APOC3 protein in the vast majority of the hepatocytes in our mouse models. And what that corresponded to when we knocked out that APOC3 protein was, especially in the hypertriglyceridemic model, reduction in triglycerides by about 95%. So you're going from a extremely hypertriglyceridemic mouse to something that's really starting to approach more of a normal range. And so we're super excited about this as an initial proof of concept. We also went through some of the safety data and just general optimization of the systems that we've done. But we're really excited about seeing where this continues to go, and are continuing to optimize and drive towards our development candidate nomination.
That's excellent. Great. I guess piggybacking on that, talk us through, what is in the foreseeable future, the next year or two for Mammoth?
Yeah. So we've had an exciting year so far. We just kicked off a partnership with Regeneron. I think, one of the things maybe I kind of vaguely alluded to earlier is the need to really integrate all the different pieces together and not just think about the CRISPR component. And so Regeneron's been doing a lot of work with retargeting capsids to different tissues, and that was really a key driver of how do we bring in all these other technologies and all this disease expertise as well. We're a 150-person biotech. It takes time to build up. There's only so much you can do at that size. And so being able to partner with them, bring in that technology and expertise, I think really has been a key thing that happened earlier this year. So, moving forward, I think there's going to be a lot of focus, of course, on pushing forward these new programs with them. So combining their new retargeted capsids with our cargo or ultracompact cargo. So really, I think it's when we started chatting with them, it really was clear this is like two puzzle pieces that just kind of click together quite perfectly. So we're really, really excited to work with them. And I think there's a lot of work that we'll be driving forward there. More generally, of course, on the program that I mentioned, to your last question on APOC3, we're driving that forward, very, very aggressively. And this will be, the first time really that these ultra-compact systems are put into the clinic. And so we're really, really excited to showcase that these systems are not only, great scientific discoveries, but are also robust editors that have therapeutic, potential. And so, we're driving that forward. And beyond that, I think there's lots more to come on the pipeline. I think that's one of the exciting things about gene editing is establish this technology that the dream or the analogy we always give is change the genome into like a Word document. And of course it's more nuanced than that. It's more challenging than that. But I think we're excited to really chip away at that problem and continuing to push forward, more innovative therapies that push that boundary just a little bit further, where to the point where we can, actually think about the genome in a different way and start to leverage these decades now of knowledge that we've figured out about the genome to be able to treat and ultimately, cure disease, right? Not just have these be, once a month treatments or things, but really create cures to patients where it's no longer a patient, but just a healthy individual that's living a normal life. So that's our dream, and maybe getting a little bit too broad, but I think ultimately that's really what we're excited about, and taking steps to build that over the course of decades is our goal.
That's wonderful. Not too broad. I think that's always the goal, and so as long as you have that, as your North Star, you're moving toward it. You're doing the right things. That's great. And I hope too soon, maybe in the next 6 months to a year, we can circle back and have you back on and talk us through where Mammoth has progressed since this conversation. So
yeah, that would be awesome. Definitely would welcome the opportunity.
Good. All right, listeners. Well, that wraps up this episode featuring Lucas Harrington of Mammoth Biosciences. Thanks to you, listeners, for your time. Check us out at cellandgene.com, and we'll talk to you soon.